Welcome to this live stream. I'm delighted this afternoon to be again with Professor James Shapiro from Chicago. How are you doing, Professor?
Fine, just a minute.
No worries.
I'm on my podcast.
Oh, okay, no problem. I talked to him. All right, okay. Bye.
Bye.
How do I say bye? Okay.
Brilliant.
I think I'm free now.
Brilliant. Okay. So, Professor Shapiro, you wrote a paper in 2020, a landmark paper called All Living Cells Are Cognitive. And this is obviously a revolutionary idea, although the seeds of this idea go back a few decades. So the first thing I'd like to ask you, Professor, is what do you mean by cognitive in this context?
What I mean by cognitive is the ability of even the simplest cells to acquire information about external or internal conditions and take actions based on that information. What I wrote was biological cognition means that cells are able to perceive changing features of their internal and external environment and undertake responses directed to survival, growth, and reproduction of themselves or their clonal relatives. So it's the essential sensory function of a living cell, and to respond to what information it receives about itself and about its surroundings.
Okay, brilliant. And one of the things which is important when you speak about cognition is that cognition is actually involved in reproduction. So one of the examples you give in one of your talks is about the proofreading system in reproduction, which is absolutely vital. And this is a cognitive system.
Yes.
Could you elaborate on this point, please?
The proofreading system involves the DNA polymerases that are doing the genome replication that they incorporate as they're copying, making strands complementary to the template. The enzymes which do the replication are called proofreading DNA polymerases, because if they replicate it, introduce an incorrect nucleotide, that changes the structure of the double strand that just is forming. And the enzyme can sense that and change its action, its activity. It becomes a nuclease, and it removes one or two or three nucleotides, and then carries on replicating afterwards, and incorporate the wrong nucleotide by about at least maybe another 10,000-fold. And that means that the ability to sense the incorrect structure of the double helix is critical to being able to execute the proofreading function. There are DNA polymerases which are not proofreading polymerases. They're called error-prone or mutator polymerases, and they're often involved in repair of DNA which has been chemically damaged.
Right. Thank you for that description, Professor. Now, when it comes to the proofreading system, this is obviously something that would indicate that the genome is not a faithful self-replicator. It cannot just replicate by itself. There needs to be an input from the cell to help it replicate. Would you say that's a correct description?
Yes, that's correct. I mean, it needs the proteins, it needs the nucleotide precursors as well. I mean, it's not just accurate transcription, it's any transcription or replication at all is dependent upon the cell. The idea that the genome is somehow independent of the cell doesn't make sense.
Right. And would you say that if the proofreading system was not present, the errors would be sufficient to cause the organism to essentially disintegrate?
Abilities to encode things accurately would be greatly compromised. Because if you have 1 in 10,000 errors, that means that many sequences will be non-functional, and the organism is unlikely to survive that. There are other proofreading mechanisms. There's something that's called mismatch repair for those rare improper incorporations which survive the proofreading system. And there, after post-replication, the genome is monitored by proteins which again sense that the helix is not properly formed. Remove the improper nucleotide or a stretch of nucleotides which include it, and replace that with properly synthesized a complementary strand. So the whole DNA replication system has basically a double backup to ensure its accuracy.
Thank you for that description, Professor. One of the things I wanted to highlight is in the famous debate between Richard Dawkins and Dennis Noble highlighted this proofreading system that you're actually referring to, and he kept referring to yourself in the debate as being one of the pioneers of this type of thinking. However, Dawkins' response was that it doesn't really take away from the gene-centric perspective. Even if this proofreading system does exist, the gene-centered perspective, genes created us, mind and body, that's unaffected. What would your response be to that?
Well, I think he's not thinking biologically. The genes are, genome is a database. It has to be properly replicated, it has to be properly read, it has to be properly interpreted. And there are some things which are not specified in the genome, such as the structure of the cell envelope. And the genome itself is not sufficient information to determine the entire reproduction or growth of an organism. It's simply, as I said, a database so that you can make the proper proteins and RNA molecules that are needed for reproduction.
Right. So in terms of causality, it does have an impact. If we go from a reductive perspective of bottom up, genes to organisms, the proofreading system itself is evidence that, we have multiple causations going in different directions, right?
Yes, that's the way Dennis likes to put it. Up and down causality. And
sorry, let me ask you a related question. I lost you there for a second. You've described the genome as not a read-only memory system, but a read-write memory system. Can you please elaborate on this point in terms of cognition?
Well, the genome is, all cells have the ability to modify their genomes, their DNA. And they do this in response to various stimuli. Sometimes it's challenges to growth. Sometimes it's other properties. Sometimes it's biologically controlled. And the genome can be changed by the cells themselves. And that's why it's a read-write memory system, not a read-only memory system. The view of the Neo-Darwinists is that the genome directs everything, but and changes only by accident. But in fact, the cells play an active role in changing their genomes. They do it in multiple ways. They have ways of rearranging the DNA, of synthesizing new DNA. And the more we learn from genome sequencing, the more amazing we find the abilities of cells to write new DNA sequences to be.
Yes. One of the things that you've written in your book, Evolution, A View from the 21st Century, the fortified version, which is much, much bigger. One of the things that you've written is that genetics have taught us about many processes that lead to biological novelties independently of natural selection. Hybridization, genome duplication.
I've lost contact with you.
Okay, I'll repeat the question, Professor. In your book, Evolution from a 21st Century Fortified, you've written the following segment. You've said that genetics have taught us about many processes that lead to biological novelties independently of natural selection. Hybridization, genome duplication, symbiogenesis, chromosome restructuring, horizontal DNA transfer, mobile genetic elements, epigenetic switches, and natural genetic engineering. Are all of these processes which are the all natural genetic engineering? Sorry, one second. Are all of these processes documented, as in they've been shown through scientific research now?
Well, that's why the second edition is so thick, because I have articles in there which include lots of references which document those statements. We have hundreds or thousands of scientific papers showing that cells change the structure of their chromosomes, that they move DNA around, that they create changes, and that these processes occur in response often to stresses that the cell or the organism experience.
Right. One of the things, Professor, that came up in the debate between Dennis Noble and Professor Richard Dawkins was genome restructuring. So the process of genome restructuring, for which Noble highlights that there's plenty of evidence. And this particular example of the genome being restructured, which was highlighted in the debate, the Nature paper of, I believe it's 2000, what we found is that there's genome restructuring happening in many different organisms, right, from mice to humans. How does this perspective challenge the overall picture in terms of Neo-Darwinism?
Well, Neo-Darwinism was a one-way causality. And it was all the information was in the genome, and it just got it was transferred to the organism and controlled its properties. But we know that regularly, we do it mostly in our immune system, where we restructure the DNA to make antibodies which can recognize all kinds of different invaders, different antigens. And that's a process of natural genetic engineering that goes on continually in our lymphoid system. there are organisms which have life phases where they rearrange their genomes, or where they form two different kinds of genomes, one for function and one for transmission and inheritance. And they're not the same structure. That happens in certain protozoa.
Right. And would you say an accurate description of this type of restructuring is like a prefabricated or Lego-like structure, which is used and to basically change the genome in a way that's much faster than through copying errors?
Yes, we know that a number of processes occur which take blocks of sequences and move and rearrange them and create new combinations, which, as you said, is Lego-like. And that's much faster than changing things slowly, one component at a time, one slightly modified component at a time. That's very, very slow. And organisms do things much faster. We know about mobile genetic elements and how they move around in the genome and that change it in that way. And as they move around, they change the control properties of the genome, and that changes phenotypic characteristics. So we know a variety of ways that the genome doesn't change one nucleotide at a time. And I was recently, or the chapter which I think I sent you,
yes,
on chromoanagenesis, this process of multiple chromosome restructurings in one cell division. And I'm trying to think of the argument. Oh,
chromoanagenesis.
Yes, I know. I'm trying to remember what it was that I was going to say. The chromoanagenesis creates large changes in the genome.
Yeah.
Well, just the fact that it restructures the genome at many places and creates many new sequence combinations is a way of modifying the functions and creating new functions.
Yes, absolutely. One of the other questions I wanted to ask you is that famous Nature paper of 2000, in which we have evidence of the chromosome of the genome being restructured, right? This is something which Dennis Noble credits you with pointing out to him, that particular paper, and this was overlooked by the majority of geneticists. So why is it that you picked up on it when the majority of the biological community just seemed to have overlooked the significance of that?
I'm not entirely sure which paper you're referring to.
This is the 2001 paper, figure 42, which is showing that. Yes.
Yeah, I know what you're referring to now. Yes, the draft human genome paper.
Yes.
In Nature. And it has a figure, two figures actually, showing that proteins evolve. Proteins are made up of segments like Legos that are called domains. And proteins evolve in part by rearranging domains and making new combinations of domains. The domains have different subfunctions, and they can create proteins with new composite functions by rearrangement. And the rearrangement can occur in a variety of different ways, but it indicates that the genome doesn't change changes. Gradually, it can change quite rapidly and create new proteins. And they're chimeras, and they have new functionalities. And I think what I was forgetting earlier when I referred to chromatogenesis was it turns out that structural changes to chromosomes in many organisms, and certainly in the our genomes, is much more frequent than our nucleotide substitutions, which, as we pointed out, are suppressed by proofreading systems. But the structural changes occur more frequently, and so they're much more likely to be useful in changing an organism's characteristics. And the frequent by certain processes, certain stresses and events. Interspecific hybridization is one of the best documented ones.
Right. And just to key off your point about mutations being suppressed, right, through the proofreading system, the standard Neo-Darwinian story is that natural selection working on random mutations explains the biological complexities that we have. Yet, according to what you're saying, randomness being the main source of... I'll repeat the question, Professor. The standard Neo-Darwinian story is that natural selection plus random mutations is enough to explain biological novelty. But according to what you're saying, randomness is not sufficient.
Randomness is not just the probabilities become vanishingly small. If you have a mutation frequency of 1 in 100, which is very high, orders of magnitude higher than in reality, by the time you have 10 a sequence 10 bases long, the probability is 10 to the minus 20. So just changing single nucleotides is not going to give you something new. Plus, which when you make these small changes that they were talking about, you modify single characters at a time. And how long do you have to wait for one character and another character and yet another character to be modified in such a way that they can work together? It doesn't make sense.
Right. And when it comes to the crystal metaphor that's actually given for the genome, right? That the genome self-replicates much like a crystal. This goes back to Schrodinger's famous book, What is Life? Do you think we should drop this crystal metaphor now that we know about the proofreading system?
Yes, and there are other systems, because it is inevitably going to be problems in the replication process. And cells are filled with repair systems to take care of those problems that arise. And proofreading is not the only cellular activity which is required to complete cell division and DNA and genome replication. There are also many repair, double-strand break repair systems and systems to make sure that the whole genome gets duplicated at each cell division.
Thank you for that answer, Professor. And now moving on to cancer, you've been involved with Professor Dennis Noble in terms of the Cancer Research Group. This research group is obviously driven by studying cancer from this new biological perspective of looking at a non-Neo-Darwinian theory in terms of how to study cancer, how to mitigate cancer. What are some of the implications for cancer research of some of the work that you and Dennis Noble have been involved in?
Well, we just helped organize a conference on it, and to treat cancer, as an evolutionary disease, and to recognize that evolution in cancer cells is subject to stimuli and changes. And what we know is, for example, there are now, I think, pretty clear evidence that many treatments like chemotherapy stimulate evolution of cancer cells, and in fact turn a perhaps non-lethal cancer into a deadly one. And there are a number of practitioners now who are using what they call adaptive therapy, where they minimize the amount of poisons they give a cancer patient because they know that they don't extend life properly, because they can trigger this increase in virulence of the tumors.
Right. Thank you for that answer, Professor. Another thing I'd like to move on to is a somatic hypermutation. So one of the things that Professor Dennis Noble pointed out in his debate with Richard Dawkins was about corona, that our body has the ability to downgrade the proofreading system in order to increase the mutation rate, so we can have new genetic sequences which can be used to actually to protect ourselves from the likes of corona. Would you like to elaborate on this point?
Well, I think that Dennis has a little bit mistaken there. Somatic hypermutation refers to the process that goes on after the antibodies are formed and are circulating in. If one of them encounters an antigen, it changes the cell that's making it, and it turns on a process called somatic hypermutation. And somatic hypermutation involves actually the action of an enzyme called cytidine activation-induced deaminase, which removes cytosines from DNA strands, and does it in a very specific way, which affects specifically the antigen binding part of the DNA, which encodes the antigen binding part of the antibody molecule. So not only is it changing the mutability of the DNA, but it's also localizing that a specific region, so that if it doesn't change the constant part of the molecule. There's a whole separate system of DNA exchange, breakage, and rejoining, which modifies the constant part of the antibody to send it to the proper tissues in the body. So the immune system is a fantastic illustration of what is possible in terms of what I call natural genetic engineering.
Right. The way that you've described it, this is the way that I've heard Professor Dennis Noble also describe it. Maybe I didn't get the exact order right. So the other thing I wanted to ask you is this particular feature that we have now understood about how the body reacts in terms of the immune system, the way that you've described it. What is the implications for this for the broader picture again? Doesn't it again show us that the one-way causation of the central dogma is incorrect?
Yes. All these processes are regulated. The cell as a whole has control over the processes. And DNA replication it starts, it ends. It can be directed. All of these things happen under the control of the cell as a whole, not just automatically. So the process of duplicating the genome and the processes of changing the genome are conditioned by or controlled by what the experiences of the cells, what has happened to them, and where they are. So the cells in the immune system, for example, have the ability to rearrange the antibody-forming DNA, but other cells in the body don't have that ability. So that's another form of higher level control.
Absolutely. Thank you for that answer, Professor. Now, another thing that I wanted to speak about was extracellular vehicles, or what were known as Darwin's gemmules, right? So this is something which challenges the Weismann barrier in terms of the input coming from the external environment and going downwards. Is this something that you recommend as a research area, and the implications for this obviously are going to be far-reaching?
I'm losing you again.
Okay, I'll repeat the question, Professor. When it comes to what's known as Darwin's gemmules, this idea that the external environment can input through these vesicles information which will help the organism develop in a different way, right? So is this something as an area of research that you would recommend in the future?
Well, there are people doing it now. It's a very active field of research. It's called transgenerational epigenetic inheritance. And so, for example, you can train a nematode worm to avoid certain bacterial species. And then you can mate it and find out that its progeny have picked up the ability to avoid that species. And we know that occurs because we know how that occurs, in part at least, because the worm produces vesicles, which are lipid bulbs, if you will, balls, and inside of them they contain some of the RNA molecules, which are directed towards, which have epigenetic effects to make certain responses to the environment, and those can be transmitted to the gametes, and then transmitted to the next generation. Inheritance as well as a DNA-based one. And the whole idea of the Weismann barrier really never made sense, because you can't have a barrier between the soma and the germline in a plant. The germline develops from the soma. So it didn't make sense, and certainly doesn't make sense for single-celled organisms.
Right. Okay. On a separate topic, I wanted to ask you about the definition of genes. So genes are sometimes defined as strands of DNA within the genome, and other times a gene is defined as anything that is inherited, right? And these sometimes are both definitions are used at the same time. How would you define a gene, and what would you say about those who define it in these two different contradictory ways?
I don't like to use the word gene. I think I mentioned George Beadle wrote a paper in the 1940s where he called the gene the basic unit of life. But a gene or a genetic locus is not a unit, it's a system. It contains pieces of coding and non-coding DNA. It contains DNA that affects its expression, that allows it to be regulated. And those different parts of the sequence can change. And you can have the same regulatory sequence, but controlling the synthesis of different proteins so that they can be what's called coordinately regulated. And that's important when the proteins have to work together. So I think that the gene is a great oversimplification. And to think of it as the basic unit of life is just an extreme form of genetic determinism.
Right.
The genome is in constant communication with the rest of the cell, with the rest of the whole organism. And they can't be separated from each other.
Right. Thank you for that answer, Professor. Another thing I'd like to ask you is, in your paper, All Living Cells Are Cognitive, you conclude there are at least 2 lessons to learn from the examples of cognition, prokaryotic cognition. The first is, evolution has succeeded in generating functional cognitive capabilities from limited cellular and molecular components. The second is, cognition is likely to prove an essential feature of all successful cellular survival, growth, and reproductions. What are the implications of these conclusions?
Let me just check my notes here. Yeah, I think the implications are that cognitive processes are basic to life as we know it. Cells couldn't survive doing all of the complicated you have to replicate the DNA with a polymerase and set up the replication system, but you have to make all the precursors. And it has to be coordinated with the rest of the metabolism. So how that's all coordinated and made to work together and does so smoothly under normal circumstances is something that we need to understand better than we do now. I think that for origin of life researchers, there's a challenge to figure out how those capabilities originated. How do cells adjust their properties so smoothly and so effectively as they do? And I think the challenge for evolution researchers is to determine what role that cognition may play in biasing the natural genetic engineering capabilities towards producing the adaptive outcomes. The Neo-Darwinists would say it's impossible to ask. But now that we know about transposable elements and multiple DNA rearrangements, which can be triggered by certain cell processes, we can ask those questions because we know we can trigger genome change, and we can then begin to ask how do conditions, outside conditions, do they play any role in changing the outcomes?
Right. Okay. Thank you for that comprehensive answer, Professor. I'd like to end upon a question about the third way of evolution. So the third way is a project that obviously involves a lot of scientists, including Professor Noble and others. The research to challenge the assumptions of Neo-Darwinism has been out for years now. Yet the trend is still that Neo-Darwinism is dominating evolutionary biology. Why do you think that's the case?
There's some change, the personnel change. The people who are still in charge of evolutionary biology were brought up on a Neo-Darwinian agenda. And they fail to recognize all the tremendous changes that have occurred. For example, Dennis Noble and I wrote a paper, Why Don't Conventional Evolutionists Tell the Whole Truth About Evolution? We know, for example, that transposable elements are an important part of evolution. They're central in changing the regulation of many different proteins, which is important in producing different organisms. They're important in providing special capabilities to cells. And the standard textbook on evolution, which is Fukuyama's, doesn't even mention Barbara McClintock, who discovered transposable elements. And that's quite extraordinary. Genomes change, and it's not even recognized in the main textbook. Maybe in the last edition, I don't know about that, but the previous one, it was missing. So I just don't think they're keeping up with all of the molecular genetics that's going on, and finding out that the assumptions that we had in the last century, in the middle of the last century, have been turned upside down. We thought that the only thing that the genome did was code for proteins. And now we know it codes for more RNA than it does for proteins, at least in complicated organisms, in complex organisms. There's so many things that have changed. We were talking about protein evolution in a Lego-like fashion. The one that Dawkins didn't know about, and that Dennis pointed out to him in the debate. That's a big change, and that doesn't get mentioned very much, because it's a much more abrupt change than conceived of in the Neo-Darwinian view. I think they just ignore it.
Right. And just speaking about the debate, what were your thoughts overall about the debate? Were you surprised that Dawkins wasn't aware of a lot of the contemporary molecular findings?
No, I think once you think you've got the truth by the tail, you stop trying to learn. And people get stuck in what McClintock calls the now explanation. That is the currently fashionable explanation. And once they get stuck there, they cease to be aware or to be able to understand or filter new things that come out which turn the thinking around. It's like the inheritance of acquired characteristics. We wouldn't have known about how it works if we didn't know about extracellular vesicles containing RNAs, which affect the epigenetic modification of the genome, but and how those processes take place. If you think that's an eternal verity, an unquestionable truth, you don't pay attention to the literature, which is outside your immediate field of expertise.
Absolutely. So thank you so much, Professor James Shapiro, for your time. Would you like to tell the viewers if there's anything else you're working on? I know you're still active in the field.
No, I'm trying to think of a way to write a general book to bring all this new information to people in a way that the general reader can understand. And I haven't yet succeeded in doing that. But I think they should be aware that there's so much going on in genomics and DNA sequencing and molecular biology and evolutionary comparisons of DNA sequences, which tells us that the processes that are going on are very different from what we all learned in school. And this is the appreciation of this is spreading in the scientific community, but it hasn't broken through to the general public yet. And I think it will be very helpful when it does, so that they can begin to learn about all these fantastic things that organisms can do, and to begin to appreciate how much organisms control their immunity, their inheritance, their DNA, and how they are capable of changing it when they have to change it. It's my belief that the ability to change is fundamental to the continuation of life, because everything will have to evolve at some point or another. And all the organisms we know about of rewriting their DNA. So that's a feature of life which I think is quite important. And if you combine that with the ability of organisms to interpret their conditions, internal and external conditions, you begin to take a very different view of how the evolutionary process could work. And there's lots of science behind both of those assertions. And I think it's going to be a very exciting time in the 21st century for our understanding of how evolution proceeds.
Absolutely, absolutely. Thank you so much, Professor James Shapiro, for giving us your time. It's been a pleasure speaking to you this afternoon.
It's been a pleasure. Thank you.
Goodbye.
Bye-bye.